लाइव में हम लोग दिख रहे हैं वहाँ पे। या सो वो तो छोड़ दीजिए ना आगे। हाँ मिस्टर श्री प्रसाद।
हाँ जी नमस्ते।
नमस्कार।
नमस्ते। हाउ आर यू?
हाँ very good, very good. So yeah, how so it has been hectic for you?
No. Today two programs. Morning one program is over. Now this second.
Sir is also here. Our director.
Good afternoon, sir. Namaste.
लेकिन जैसे ही इसको हटाऊंगा वैसे तो वो निकल जाएगा, छोटा हो जाएगा।
So I can just use the microphone because I don't have anything to display, right?
Yes sir, yes sir.
Okay.
Because searching is able to join. He says I'm clicking it, copy pasting it, but still there is a problem.
Okay.
So मैंने तो किया.
No, I think you have to log into Google first, Gmail first, then only it will work. Google Meet, you have to get into your Gmail first.
पहले जीमेल में आ लेना पड़ेगा, गूगल में आना पड़ेगा। वही तो मैं तू बोले कि वो चला जाएगा।
मुझे जस्ट नहीं करते, मैं वहां से
अभी तो स्टार्ट हो जाएगा। ठीक है, छोड़ दीजिए।
जब मुझे एंट्री कर दो।
So I think Sandeep, you will start and then you will invite me.
Yes, sir.
Okay.
Yeah. Where is your share? Agenda. ये कौन है? कौन? Mr. Sri Prasad. This Madam Rita Bhattacharya has joined from Viva.
Yes.
Namaste, Madam.
Namaskar.
She is our National Secretary of our Women's Wing.
Okay, Namaskar, Madam. Madam, our Director Sir is also here.
Namaskar.
Namaskar, Director Sir.
Namaskar, Ritaji. Yeah, I mean, it's. I wish we could have a physical meeting here, but anyway, we'll just go with the whatever is possible.
Yes.
Yeah. Okay, Sandeep, you can start. Yeah, you can start.
Yeah, Dr. Sauche has joined.
Okay.
So I can start. Good afternoon, everyone. So I welcome on behalf of National Institute of Biomedical Genomics, all the faculty members and members of NIBMG. I welcome all of you to our outreach program, which is the part of this India International Science Festival Vigyan Yatra. This is without any further any delay, I would like to request Professor Soumitra Das to give his opening remarks and start this program.
Welcome you all. Actually, it's a great pleasure to give the opening remarks in this Vigyan Yatra. As you all know, last 4 years, we have been doing it at the National Institute of Biomedical Genomics. And, we were also looking forward to do a physical meeting this year as well. But actually, situation somehow prevented us, and we are trying to go it online. But in a way, it is actually blessings in disguise because we are getting more people accommodated across India to have this kind of an online meeting so that they can all participate. As you know, India International Science Festival has been going on for the last 4 years. Well, I think it's more than that. And Vigyan Yatra is an integral part of that. And where we try to impress upon the students, we try to showcase the technology that we are actually doing it in the institute. Past few years, actually, several students visited us and in our institute, and we showed them the different facilities that we have it. We have also actually Shobik Mukhopadhyay, Professor Arindam Maitra, Dr. Alpana Dave, all of them actually put together a very interesting and exciting program today. And I'm very happy that actually this year we have picked up microbiome and the genomics interlink. This National Institute of Biomedical Genomics is one such institute, an unique institute in the country, where our mandate is to understand the importance of biomedical genomics in the human health. And essentially, we try to understand the genomics, and we want to practice genomics so that it is useful for taking care of the human health problems, which is the human health burden. We work on cancers, we work on infectious disease, we work on chronic disease, and everything is actually somehow integrated with the computational biology and statistical biology, statistical analysis. And now lately, we have been actually interested to try to understand that how the microbiome and the environment also influences the genetic determinant. Previously, we used to think that there are a lot of hereditary component in all the disease, and that is actually a major burden. But now it looks like not only the hereditary component, it can be actually because of the changes in the microbiome and actually changes in the epigenetic changes because of the microenvironment can actually equally be responsible for triggering into disease. Now, while saying that, I essentially tried to say that staying in the National Institute of Biomedical Genomics, now we are increasingly interested to understand how the microbiome in a patient can actually link it up and influence the genomics. And today we have none other than the pioneer of the microbiome research in the country, Professor Jogesh Shouche. I'm so glad, Jogeshji, that you have actually agreed. I know you are very busy. And we also have a youngster, Shobhik Mukhopadhyay. He's actually doing the microbiome and genomics and linking them together. So they will have actually their first-hand experience sharing with you. And at the same time, we'll also try to give you a tour of the institute with the help of our institute faculty so that you don't miss anything. So, I mean, that the positive spirit is essentially we move ahead, even if we have some hurdles coming on the way. But that's how Dr. Bhattacharya was saying, Dr. Rita Bhattacharya was saying that, we have to overcome the situation, and we are overcoming the situation. And I think we'll just try to see how we can move ahead. And I'm sure IISF will be a great success like past few years. We'll have it in a different mode this time, but I think it will probably attract more attention. Not only nationally, it can be stretched to globally. I think without much ado, I think I would like to move to Dr. Sandeep Singh to conduct this meeting. And I'm again thankful to all of you, from the Vigyan Yatra organizers, the IISF coordinators, Dr. Bhattacharya, and my faculty colleagues, so that, and I'm sure all the students, in the institute, we encourage students for short-term training. We have some long-term training. So you explore it. The moment the COVID situation is better, we'll try to invite you guys to come to the campus and enjoy those things firsthand. But pending to that, at least we'll try to have a glimpse of it. And essentially, that's all is today's program. And with all the best wishes, Dr. Sandeep Singh, over to you. Thank you. Namaskar.
Thank you very much, sir, for your opening remarks. And to take this program further, we have a short video of India International Science Festival. So we would like to play that video, and you all watch this video.
India International Science Festival, or IISF, is a much-awaited annual event celebrating India's achievements in the fields of science, technology and innovation. The objective of IISF is to spread scientific awareness among all sections of society. IISF provides a platform to students, teachers, researchers, innovators, scientists, craftsmen, policymakers and to all the sections of the society to deliberate on the development of the society through science. IISF is being organized since 2015. So far, 5 IISFs have been organized. This year, the 6th edition of the India International Science Festival IISF is being organized in a virtual format from 22nd to 25th December 2020.
लोगों के अंदर एक ऐसी समझ है कि विज्ञान एक बहुत ही कठिन, गंभीर इस प्रकार का विषय है। इसलिए हमने ऐसे सोचा कि विज्ञान को एक उत्सव के रूप में प्रस्तुत करना चाहिए, विज्ञान का उत्सव मनाना चाहिए। इसलिए हमने ये सोचा कि ये भारत का अंतर्राष्ट्रीय विज्ञान महोत्सव इस प्रकार का ये कार्यक्रम विकसित करेंगे।
On November 17th, the curtain raiser of the 6th India International Science Festival was organized in New Delhi. The program was inaugurated by Union Minister of Science and Technology, Earth Sciences and Health and Family Welfare, Dr. Harshvardhan, in the presence of all the officials of the concerned ministries and departments. Adhering to the COVID-19 guidelines, this year the Mega Festival of Science will be held in a virtual format. The theme of this festival, which will run from the 22nd to 25th December, is Science for Atmanirbhar Bharat and Global Welfare. During the curtain raiser of the festival, website of IISF 2020 and a brochure of the mega event were also launched.
सारा देश जानता है कि 2015 से लगातार हम हर वर्ष देश में इंडिया इंटरनेशनल साइंस फेस्टिवल आयोजित करते हैं। ये हमारा छठा साइंस फेस्टिवल है। विज्ञान को कैसे एक फेस्टिवल के रूप में, एक आंदोलन के रूप में देश में विकसित किया जाए, क्योंकि हमारे वैज्ञानिकों का भी यही मत है और यही प्रयास है कि विज्ञान के माध्यम से जनता की सब अनसुलझी समस्याओं का समाधान किया जाए।
The responsibility to organize this mega science festival in collaboration with Vigyan Bharati and all the concerned ministries and departments of Government of India has been given to the Council of Scientific and Industrial Research, CSIR.
We take science to people in the language that they understand it. See, general public is very curious about what happens in our laboratories and all, and they hardly get to know what scientists behind the walls of laboratories actually do. And even if the general public tries to understand it, for many people is beyond their comprehension. But IISF is one such platform on which we tell people in their own language what happens and all, so that people feel excited and feel involved, that why is society supporting so much of science and technology. So that's one of the major purposes of IISF.
The nodal institution for organizing the 6th IISF is CSIR's National Institute of Science, Technology and Development Studies, that is CSIR, NISTADS, based in New Delhi.
We have come up with new events. 13 new events have been added this time. And they are, I will say, need of the hour. For example, sustainable development goals, our environment, how masses should be made aware about the sensitivity towards environment. So we have created a whole lot of events on water, energy.
During IISF 2020, 41 events will be organized on various aspects of science and technology, highlighting the role of Indian science and fight against the current pandemic. Several new events have been added based on relevant issues like history of Indian science, philosophy and science, agri-tech, clean air, energy, waste and sanitation, biodiversity and science diplomacy. As the festival is being organized on a virtual platform, so the reach of the festival is expected to expand further. It is assumed that IISF 2020 will attract more than 1 lakh participants from India and abroad. The Grand Festival of Science will once again provide people from all walks of life a unique platform to share and celebrate the advancements of science, technology and innovation. To know about the various events during IISF 2020, you can visit the website of IISF 2020 or NISTADS. You can also register online to participate in these programs. All of you are requested to participate in these programs in large numbers.
So, after viewing this video, I hope you all have understood the mission and vision of India International Science Festival. And to take it further with the India International Science Festival ethos, NIBMG has organized this Vigyan Yatra, where we are going to introduce you with the facilities which NIBMG is having to do the research in the field of genomics, which Dr. Souvik Mukherjee will be introducing to you. But before that, it is my honor to introduce you our guest speaker, Professor Yogesh Sauche. Professor Sauche is the scientist emeritus at National Center for Cell Science, Pune. He has more than he has more. Let me see. So he has more than 30 years of research experience in the field of molecular taxonomy and biodiversity of microbes. Dr. Sauche has more than 300 publications in his credits in the field of systematic and evolutionary microbiology and microbial ecology. His work is cited more than 11,000 times by scientific fraternity. So it is, without any doubt, I can say that there could not be anybody more eligible than Dr. Sauche to introduce to you the field of microbiome research and the area and the fascination which is there with this research field. So without any further ado, may I request sir to please deliver his lecture, and we all are eager to hear from you, sir. So kindly share your slide. Yeah, thank you, sir. Sir, kindly unmute.
And make it full screen.
Sir, it got, yeah. Your sir, your mic is getting muted all of a sudden. Maybe if you log in.
Yeah, I think Yogesh, you can log out and log in again.
Please bear with us. There is some technical issues. Dr. Sauche will be online soon.
So Sandeep, you make him presenter.
I think your mic is not working. So do you have any?
Yeah, now okay. Now just see. Can you hear me now, right?
No, sir, it is not audible.
Yeah. Okay.
Share your screen, Javesh. Share your screen.
Am I audible now?
Yeah, I can see.
Yeah, you are audible.
Okay, good. Yeah, that's fine then. Now the problem is resolved now.
But
Right, fine. Yeah.
show the
Yeah,
slide.
now I'm doing it. Yeah. Just 1 second. Yeah. Sorry about that. And
Yes, sir. I can. Yeah, now you can hear it.
now you can see the screen also. Can you see the screen?
Yeah, we can see it. Make it full screen.
It is full screen only from at my end.
No, I think it's a single slide. It is a slide view.
Okay. Now what I'll do is then. Now you can see the full slide.
Yes.
Okay, so I think
Yes.
it's always a problem.
Please go on the full screen.
It is. I think this is the. If I go full screen, it is getting into some other problem. So
No problem. This is fine.
So I think we'll go it this. Do it this way.
Go
only.
ahead. Yeah. Go ahead.
Yeah. So fine. Sorry for that confusion, but now I think we are more or less. And thank you very much for this nice opportunity to discuss with you about the human microbiome. I would like to thank Director NIBMG for giving me this opportunity and Sandeep for his nice words of introduction. So today we are going to talk about microbiome, what is microbiome and why it is important for us. Before we understand what the microbiome is, we should know what the microbes are, because many of you would not know what the microbes are. So microbes are basically small living organisms. They are so tiny that you cannot see them with your eyes, and you need either a simple light microscope or you need electron microscope to see them. And how small they are. So there are few microns. And what is a micron? Micron is 1,000 part of a millimeter. Millimeter, of course, everybody would know because you use the scales in schools, and those scales, the smallest unit you have is the millimeter. So microbes are 1,000 times smaller than the smallest scale, smallest unit which you see on your scale. So they are that small, and that's why you need a specialized instrument like microscope to see them. Viruses are still smaller, and you need electron microscope to see them. So if you quickly have a look over here, you will see that these are some of the things which pollens, very tiny pollens are visible to our eyes, but beyond that, these things are not visible to our eyes, and most of the microbes, including bacteria, fall here and viruses would fall here. So how did we come to know about the microbes? So it was something like 300 years back, a person called Leeuwenhoek, he developed this very small handy instrument, and here by placing his samples here, he observed them under the microscope. He took everything that was possible. He took tooth scrapings, he took river water, well water, sediments, and he observed different life forms moving, dancing, jumping, and he called them animalcules. and he drew nice pictures of them. So this was 300 years back, and this can be called as birth of microbiology. So here he was able to observe them only under the microscope, and he could just describe various shapes. But in order to study them in little more details, you need to have them in larger quantity. So methodology for growing them in larger quantity was developed much later. Something around 900, a person called Vigyan developed laboratory media in which the microbes can be grown. So you can see here microbes growing on the plates, and that kind of exploded the information about the microbes, and we came to know a lot more about microbes that are present everywhere. And then it was a kind of explosion of information about microbes. So what we know is it is a staggering number. You can see here these many, on 5 there are so many zeros. Those many number of microbes are known to be exist on the earth, and they are present everywhere. You cannot imagine a place where microbes are not present. As I said earlier, they are present in water, they are present in soil, they are present in rocks where we cannot imagine any life, they are present at deep oceans, they are present below the earth's surface, they are present in boiling hot water springs, they are present in sub-zero temperatures, they are present at acidic pH, they are present at alkaline pH, and they are present at places where we cannot imagine a life. We all need oxygen for our life. Without oxygen, we will die. But microbes exist in places where there is total lack of oxygen. These are called as anaerobic environments. So point is that the microbes present everywhere. And of course, the most important location of microbes is every living organism has large number of microbes associated with them. You can imagine any living organisms, may it be plant or animals, insects, marine organisms, birds. Each and every living organism has certain specific microbes associated with that, and human beings are not exception to that. Human beings also have a large number of microbes associated with that. So if you look at the microbial diversity, the number of microbes that are present, you can see that when we talk about two major categories of microbes, fungi, so far we know 75,000 different species of fungi, but estimated number is something like 200,000 to 10 million. About bacteria, estimated described species is something around 5,000, and what is estimated is 30,000 to 3 million species of bacteria. We know very little about them. So why is that we know very
Sorry about this problem. Looks like Dr. Sauche is offline. So we are trying to bring him online soon.
Oh.
It seems that there is severe technical glitch here, so that is why Dr. Sauche has some problem in joining us. We are trying to get him online, but in the meantime, I can request Dr. Sauvik Mukherjee to begin his talk. And Dr. Sauvik Mukherjee is the assistant professor at National Institute of Biomedical Genomics, and he is very much involved in the research in the field of microbiome, and he is the expert from NIBMG who can introduce you to the field, and also the research facility which is there at NIBMG to do this kind of research. So I request Dr. Sauvik Mukherjee to begin his talk, and in the meantime we are trying to bring Dr. Sauche also online. Thank you.
Okay, hi. Just I need a few minutes, 2-3 minutes.
Yes, Dr. Sauvik, you can actually initiate you can share your slides. Yeah, just select the. Yeah, go.
Yeah, has the slides been shared? Can you see it?
No, no, not yet. So we can see the entire screen. You can
Okay,
put,
okay.
yeah. Now you go to, yes, yes, yes. Go to the full screen. Yes.
Now can you
Yes,
see?
we can see now. We can see now. You can start.
Okay. So thank you for this opportunity in speaking about human microbiome research in IISF 2020. We are a new institute. New means we are just 10 years old. And as our director sir has already spoken about it, our institute is working on biomedical genomics. So we are an institute which is focused on the genomic underpinnings of anything that is in a medical research in humans on diseases. So my area of interest is understanding the host microbiome interactions in some of the diseases in humans, and I will say you through these popular examples of host microbiome interactions in humans. So before that, I would like to impress upon the fact that why are we studying the human microbiome? The reason being, we are trying to find out the missing heritability of complex kinds of disorders. Previously, we all knew that there is a hypothesis called CDCV, Common Disease Common Variant Hypothesis. And during that time, we believed that if a disease is common globally, the variants associated with it also be common. So we did a lot of studies of candidate gene and genome-wide association studies, but we could not still explain the entire missing heritability of these type of ailments. Then came the era of common disease rare variant hypothesis, where it was believed that although the diseases are common globally, yet the variants can be rare and they can be ethnicity based or population based local adaptations. We did NGS, the new kind of sequencing in which we could identify both the common and rare variants. Yet we are still struggling to find out to explain the entire heritability of these types of complex ailments. Given the fact that if you can see, other than the Mendelian kind of diseases, the complex diseases, they have this genotype to phenotype correlation. It's not a 1:1 correlation. There are different subphenotypes and there are environmental aspects also. Given this fact, we should also study other genes along with the environment and the subphenotypes to find out the associated factors related to a disease. Given this idea, the new era is now that the human microbiome is actually the solution to this missing heritability problem, and a new kind of studies are coming up that is called MWAS. That is, previously it was GWAS, genome-wide association studies. Now it is called MWAS. We're integrating both the host genome and the microbiome. We call it metagenome-wide association studies. So who coined the term microbiome? This term is coined by none other than Joshua Lederberg, who told that the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space and have been all but ignored as determinants of health and disease. And to know who is Joshua Lederberg, you should acknowledge the fact that he is the one who discovered conjugation and transduction. And therefore, he was awarded the Nobel Prize in Physiology or Medicine in 1958. Microbiome constitutes, believe it or not, 90% of your number of cells in your body. This was the idea when this field of microbiome first was introduced, and this formed the basis of the Human Microbiome Project. The idea was that the volume of the alimentary canal is 1000 ml. And if the density of bacteria is 10 to the power 11 each gram of the stool, then we can identify the total number of microbial cells to be 10 to the power 14. And thus, these are some of the studies done in 1970s that have estimated this particular ratio. But recently, it has now been changed, and now it is known that the microbiome in our human body constitutes 50% of our cells and not 90%. And how did they arrive at it? So now, based on the technological advances, it is known that the volume of the alimentary canal is not 1000 ml, but it is actually 400 ml. So based on that and keeping the density of the bacteria in the stool, we can now identify the equivalent numbers of microbial cells, 3.6 into 10 to the power 13, and this is the equivalent number of host cells present in us. So like that, there are several studies that have found out that the bacteria is to human ratio is nearly something like 1:1.3 to 2.2, so it is 1:2. So 50% of our cells are bacterial. Microbiome is present across different body regions, and previously it was thought that the intestinal microbiome is the major player, but now as the advances in the sequencing and other kinds of techniques are happening, we can find out microbiome in different parts of the human body, in the skin, in the oral cavity, in the ocular region, nasal microbiome, vaginal microbiome is now a major factor in studying the maternal health. By microbiome, we not only mean the bacteria, we also mean the fungus, the archaea, and several other kinds of organisms present. And of late, there are examples that our individual genetic background, the food we eat, and the microbiome in our body, they all interact with each other to provide us a healthy phenotype or a diseased phenotype. There are two kinds of studies in microbiome. So one is, if we want to understand who are there, what are the microbes that are present in this particular part of my body. By that, we can only do 16S amplicon sequencing. As we know, 16S is a unique gene that is present in the bacteria and not present in humans. So this gene, if we sequence, we can get what are both the culturable and the unculturable bacteria present. And for fungus, we can do 18S. So this is a kind of study where we are doing amplicon sequencing, where we are taking a single gene as a marker of that particular family, and we are sequencing that. But they will not give us the idea of why are they there? Why are these bacteria present there? What are the functional interactions they are taking place? For that, we have to do whole genome sequencing of the entire microbiome. So as I have told, 16S gene and the whole genome. So 16S, as you can see, it has got different constant and variable regions. C1, C2, C3, these are all constant regions, and V1, V2, V3, these are all variable regions. I'll not go into the gory details of the analysis, but please understand that we can make primers, universal primers on the constant regions so that all the bacteria, both known and unknown, can be sequenced. And based on the alignment of the variable regions, 97% if they are similar, then they come to the same genus. If they are similar, 99%, they come to the same species and so on. And by whole genome shotgun sequencing, we can not only identify what are there, we can also identify what are their gene families and what are their pathways present there. So while we do the sequencing, after doing the sequencing of 16S, when we analyze them, we take a unique approach. It's called the OTU approach. OTUs are called operational taxonomic units. Here, when we get all the sequences present, these sequences are then, based on their similarity, are they 97% similar or 98% similar? We can put them into separate entities, and these entities are called OTUs. Afterwards, we can then only take out a single sequence from each of these entities and align them with the microbial databases, and they give us the estimate of what are the bacteria present. This saves us more significant number of computational energy and time. And I will show you an example of one real OTU table that are present in our study. This is the OTU, and you can see these are the OTU IDs. These are the names of the bacteria up to the genus or the species levels, and this S1, S2, and S3 are the individuals. And you can see absolute number of reads. These are actually DNA sequence reads present of each of these bacteria in one individual. As you can see, total number of reads for each individual are not same. So to compare across individuals, we have to normalize them. For that, we have taken a unique approach of estimating the relative abundance. The relative abundance of a particular bacteria is the number of reads of that particular bacteria and divided by the total number of reads for that particular individual into 100. As you can well understand from there, we can identify by this sequencing experiment a very rare kind of bacteria also, which is only present 0.06%. This could not have been identified if we only do bacterial culture. So there are some of the indexes by name Shannon index and Chow index by which we can also measure the bacterial diversity within a particular region. The first study that was taken, as I have told, is the Human Microbiome Project, and it was taken up by NIH, JCVI, and several other institutes together. Their main idea was to identify the normal healthy volunteers. What are the microbiome present in various different areas of the human body? They took 250 individuals for that, and they tried to find out the baseline reference microbiome. And here, after that, when they started this study, so this was the paper that was there when they started the study, and they gave us the detail of their study designs and everything, the importance of their study. After that, when the study was being carried on, several such seminal articles came in Science and Nature, where they actually understood, where they actually showed us what is the healthy human microbiome, by which we can compare our some clinical phenotypes with that, and that can serve as the healthy. After that, an upsurge in the microbiome studies have taken place. You name a disease, and there are certain studies that have found that not only the human genome, but the microbiome is also important there. Several kinds of autoimmune diseases, diabetes, cardiovascular, and different kinds of diseases. But there was a caveat that the healthy microbiome is not found to be same for all. They have done some human microbiome project in Europe and USA. Now they wanted to say that that is the healthy baseline based on which each and every researcher in their own country can align with them of the clinical conditions and find out the alterations. There was a study at that time that came out that if you go to Africa and take the Hadza hunter-gatherers, you study their microbiome from their stool, you don't find the healthy microbiome to be similar of what is obtained in the human microbiome project from the Italians or from the other kinds of Europeans or Americans. Quite surprisingly, Bifidobacterium, that is now considered to be a marker of healthy intestine, is not present in them, completely absent in them. They have got higher levels of Prevotella, Treponema, all those kinds of microbes in their intestine, which are still now considered to be very harmful in our intestine. They are found to be associated with different kinds of diseases like IBD and several kinds of complex type of ailments. So why are these individuals having these supposedly harmful microbes in their intestine, and they are not suffering from these kinds of ailments? So then an eye-opener happened, and we found that the healthy microbiome may not be same for all. For each of the country, for each of the geographical areas, for each of the environment, food habits, we have to first find out what is our healthy human microbiome. Only then we will be able to study several kinds of clinical conditions. And why it was so? Why their microbiome was full of these supposedly harmful kind of bacteria? Because when they did a further study of the products produced by these bacteria, it is called the metabolomic study, they found that these Hadza hunter-gatherers, they actually have a foraging food habit. Their food habits are not similar to us, and therefore the bacteria present in them, Treponema and Prevotella, can digest these kinds of food products that they are consuming, which are not required in us because we are mostly consuming different kinds of foods. So I will give you a brief outlook of what we do in NIBMG in our lab, and then I will show you what others in NIBMG also do. So we are mostly, focused on skin diseases or skin microbiome and chronic wounds. So we have some projects with industry, and we also have some projects funded by DBT and DST and other kinds of funding agencies. So our major focus is on skin microbiome, and we have found out the first human skin microbiome in India, and that was the first study from India, where we have found that how the sebum and hydration levels in specific regions of the human face, they significantly predict the nature and diversity of this facial skin microbiome. Our study has shown, when we compare it with the healthy skin microbiome from Europe, we have found that proteobacteria is significantly higher in our study, and which is actually covered by this genus Streptococcus, and Corynebacterium is higher in the Europeans. And Streptococcus is considered to be harmful in the skin, and Corynebacterium is actually a beneficial bacteria. We have done till the species level, and we have found that the Streptococcus species that we have is a more of a harmful kind, considered to be harmful in Europe. And the Corynebacterium species is actually hydrolyzing your sweat. So it is hydrolyzing the sweat urea and producing the NH3 and N2. So it is a kind of a nitrogen fixing bacteria. This was the first Asian or Indian skin microbiome study, and till date it has received 50 citations. We then came on to study a skin disease, and that was called atopic eczema, or we call it in a scientific name atopic dermatitis. We needed to study the host microbiome interactions in them, not only the changes in the skin microbiome. I will be very short here that we have found, we have done here shotgun sequencing, because we did not also wanted to find which microbes are there. We also wanted to know what are the microbial gene families there, how are they functionally active. We found S. aureus to be present in the cases and S. hominis to be present in the controls. And we did a particular gene is there, filaggrin. This particular gene is a skin cornification gene, and it actually has a protein that goes and attaches with the collagen in the skin and gives the skin integrity. We found that these are some of the 40 loss of function mutations in filaggrin, which were present in different parts of the world associated with this atopic eczema. Quite intriguingly, we did not find any of them associated in India. We found some other kinds of SNPs here. Based on those other kinds of SNPs, we tried to do some haplotype-based analysis. So what a haplotype is a haploid genotype. So this is the reference unmuted haplotype, and this one is the mutant haplotype. When you take the mutant haplotype, means all of these SNPs are having some mutations here. That is a more damaging kind of a haplotype. We found some specific kind of genus or species, they are changing with the presence or absence of these filaggrin genotypes. I'll not go into the gory details of it. And we also found the microbial pathways. We found that in the cases and controls, different kinds of microbial pathways are enriched. The histidine is the amino acid, L-histidine degradation, arginine proline degradation. These kinds of gene families are more enriched in the cases, and we found that produce ammonia, and they increase your skin pH. Thus, the antimicrobial peptides are becoming inactive and becoming more and more harmful for the harmful bacteria to come. I'll skip these slides and I'll go to the model. So what we have proposed is that the filaggrin actually transforms to trans-uroconic acid that acts as a sunscreen and protects our body from the UV rays. Now, urocanase is a particular enzyme that is only present in S. aureus and absent in humans. So S. aureus in the histidine metabolism or arginine metabolism pathways, they produce this urocanase, which actually degrades this trans-uroconic acid in your skin. Thus, your skin becomes dry and it becomes harmful for eczema to happen. Some other seminal studies are there that has been done with the monozygotic and dizygotic pairs. What they have done, this is a study of the UK twins, and they have done host DNA genotyping and also 16S gene sequencing from stool samples. What they have found is the monozygotics one have a more similar kind of their gut microbiome than the dizygotic ones. And if you can see here the inter-individual variability, we have several estimates by which we can understand inter-individual variability within the monozygotic or the dizygotic or the unrelated individuals. We have found the monozygotics always have a lower microbial diversity than the other ones. So monozygotics are more similar to each other. With this, they have also seen that those individuals who have an inactive LCT gene, they have in their stool more of the bifidobacterium that can help in the breakdown of the lactose. And those who are having active LCT, in their stool samples, bifidobacterium is not so high. So this is, we all know about this particular thing, that lactose is broken down to glucose and galactose until it reaches the colon. Otherwise, if you don't break up lactose to glucose and galactose, if the lactose continues to go to the colon, most of the time abdominal pain and other kinds of things happens due to the microbiome acting on them. So this is a classic example of the host microbiome interactions for the advantage of the host. One particular SNP is there, one particular single nucleotide variation in the LCT gene that makes this protein lactase inactive. They have found those who have that particular mutant allele in them, the bifidobacterium is significantly higher. And they have checked in both winter and summer. In both the cases, it is same. So there is no temporal variation in this microbiome abundance also. I can go on and on, but I will not go in more details. This is another example that I can show of IBD. In IBD, they have also shown that there is a gene called NOD2, where there are some of the SNPs associated with IBD. They have also done their 16S gene sequencing. And what they have found is among these 163 SNPs, for 49 SNPs across the whole genome are found to be associated with the 16S microbiome abundance. And when they do some enrichment analysis of those 49 SNPs, they find mostly those genes that are present in the immunity pathways. So it is a classic example of that how host genome and microbiome is interacting together. So lastly, this is a model that now has been proposed that host genome can influence the host phenotype. Microbiome can also influence the host phenotype, and they can also interact, and that interaction also can regulate this clinical phenotypes. Some environmental factors can also play a part. They can also influence both your microbiome, like the food you eat and many other kinds of things, and the host gene regulation can also be impacted upon like that. This is a time of COVID. So in the time of COVID, I would just share some slides where there is a connection of the lung-gut axis in respiratory viral infections. It has been found that any changes in the microbiome of gut can this through the mesenteric lymphatic system, the products of those bacteria can actually travel to your pulmonary cavity, and these in the pulmonary region they can create havoc. And other than that, if the lung is also taken up by some of the pathogenic bacteria, they can also their products can also travel to your intestine, and that can trigger the production of pro-inflammatory cytokines. And this is a study which has shown the alterations of the microbiome in patients with coronavirus. And the study design is not so good. They have also taken a small number of samples, but please acknowledge the fact this is a study done during the pandemic. It's not that they have got enough amount of time to do the study design and all. What they have seen in this COVID-19 patients and the H1N1, the classic influenza patient, that is a positive control kind of thing, and a healthy controls are also there. They have found that the inter-individual variability or the diversity is actually very much higher in the controls. After that, it is in COVID-19 patients, and they are more similar in the H1N1. When they did some statistical analysis, they found the gut microbiome of the COVID-19 patients are dominated by opportunistic pathogens like Streptococcus, Rotia, Velionella, and the commensals are not there. So this is the study. They did just an observational study. They did not do any functional thing. But there are some studies that are now going on globally, and we are also a part of one of those studies where we are studying the microbiome of both the severe and the asymptomatic kinds of COVID patients, along with their genome. So coming back to our institute, now our institute has got 4 different arms. One is, of course, cancer, the gene signature and the functional genomics of cancer. We study most of the cancers present in humans, mostly oral cancer, cervical cancer, breast cancer, pancreatic cancer. We also have a separate wing for studying the host genomics along with the infectious pathogen in the infectious kind of diseases, where we study sepsis, where we study different kinds of RNA viruses, intracellular protozoans, tuberculosis and all. We have another arm of multi-omics, determinants of complex kind of disorders, where we study autoimmune skin disorders, some complications in neurodegeneration, fatty liver, preterm, and all those kinds of stuff. All these 3 groups, they have to do big data analysis. For that, we have a separate arm called statistical and computational genomics, and they also study population structure, human evolution, those kinds of things. In some few slides, I will now show you the labs that are there, where our students and our researchers can go and work. So this is the genomics facility of NIBMG. We have a state-of-the-art genomics facility. We have all the NGS instrument present, and one of the largest one is the NovaSeq that is also present in our institute. And we have generated lots and lots of gigs of data, and which is equivalent to more than 42,000 human genomes. And we also have a proteomics facility now, where we can study the proteomics also. So it's a unique institute where we have a genomics expertise, you have a proteomics expertise. A new field of science called proteogenomics is coming up, and we can study it there. We also have a facility where we can assay cytokines, more than 400 of them, for inflammation, disease, cancer, and whatnot at a given point of time. We have a flow cytometry and cell sorting facility, where we can separate the cells based on their unique characteristics, and 16 different kinds of unique features. Single cells also can be sorted in this manner. NIBMG has one of the largest dry labs facility, where we have computational labs and computational facilities with a huge amount of computational power. For functional validation, we have a zebrafish facility, because zebrafish, as you know, shares more than 70% of the genes with humans, and they lay hundreds of eggs, and the embryos are nearly transparent. They're also robust and cheap to maintain and fast to grow. So we are using this as a model organism for most of our functional validations of the genomic underpinnings. We have a confocal imaging facility, where we take all these images of our functional things that are done. Other than that, we also have a national genomics core. This is a unique enterprise where others from outside, the researchers from outside, can also use our high throughput facilities. And we have a separate group of employees for them, and you can approach them. This is the mail ID, as you can see. You can approach them, and they will help you in doing the high throughput sequencing, data analysis, and many more. We have a unique human genome hall. You can see these are the 2 individuals, Watson and Crick, and this is a Nobel laureate who came and visited us. And we have a genome hall. In this genome hall, we have 3D structures of all the biological phenomena, all the biological structures, protein structures, and the interactive kinds of things which are unique for school and college students. Last but not the least, we are now planning for a host microbiome research facility, where we not only do the sequencing part, but we also use this lab to do some anaerobic cultures, to do some immunomodulatory effects, host microbiome in vitro co-cultures and expand our horizon where we can isolate the microbial DNA and RNA from different kinds of specimens. Our microbiome, field, we have hosted a India EMBO Symposium, a conference in November. That was the last one where we had physical conferences in the era, and we had 15 international speakers and 9 national speakers. Professor Shouche also adorned the chair, and we have more than 50 international and national participants. It was organized by us here, me and Professor Partho Majumdar, who is the founder of this institute. We organized it from here, and 3 of the individuals, prominent scientists, the president of the JCVI USA, Karen Nelson, she was there, and her team, they also helped us to organize this. This was a welcome DBT and EMBO conference. And this is some of the glimpses of our director, sir, inaugurating the conference, and Professor Majumdar also delivering his lecture. There were interactive sessions, and we came up with different ideas of how to carry forward the human microbiome research in India in future. I would like to acknowledge our director, Professor Soumitra Das, for giving me the opportunity to speak in this forum. Professor Partho Majumdar for all the help that he is keeping on, encouraging me in doing this type of science. All the faculty members of NIBMG to have contributed slides on the different facilities, all the NIBMG staffs and all my lab members. Our campus is not only doing science, we are also very vibrant. We have, playgrounds, we have ponds, we do cultural programs, and we will request you to visit our institute soon in the post-corona era. Thank you, and it's a thank you from our entire NIBMG family.
Thank you very much, Dr. Souvik. It was enlightening talk. We all understood that how much important is our genome to the type of microbiome we carry in our body. And thank you also for showing the facilities which NIBMG is having and what kind of microbiome research we are all carrying out. So I'm very happy that Dr. Shouche could join us, and yes. I'm very sorry that there was some technical problem by which
Sandeep, can I continue from where it was disconnected, or
yes,
you have wound it up?
Yes, sir. Yes, sir. Please. Yeah, you can actually
put a pause button for you.
Thank you. Actually, I did not realize that I have been disconnected. So only after this thing was over, I saw that there was a message. Anyway.
Sorry, sir. Sorry very much.
No, it's okay. Yeah, these online things have their own disadvantages. There are good respect, but there are some disadvantages of that also. Yeah. So now I guess my slide is also visible. So
yes, sir.
Yeah. So I think I was disconnected somewhere here when I was talking about the use of ammonia oxidizing bacteria as a. So what people have done is, just to summarize, that from the soil, ammonia oxidizing bacteria were isolated, and they become a part of this spray. And when you use this spray, then the ammonia from your sweat is degraded by this bacteria, and you don't smell. So, and the person who developed, he met one of my colleagues, and he claimed that he uses the spray, and when he met her, he had not taken bath for a month. So the spray works that effectively. Same way, many celebrities inject Botox on their face to relax the muscles and suppress the wrinkles. But now it is being replaced by microbiome. There is this commercial skin microbiome preparation that has come up in the market, a probiotic anti-aging preparation, which does the same job as Botox. So it is environmental friendly, it is probiotic, and it is good skin microbes. The same way microbiome is linked to the intelligence also. People say that it is linked to your IQ, and those with higher IQ have a different microbiome. It is also increased to the link to the span of life, and it is shown that those who live long have a different set of microbes. So considering all these, the microbiome is a globally very lucrative business, and it is estimated that in 2024 in US alone it will be something like 1,360.49 million dollar business, and many youngsters are interested in getting into this. So this business is going to be for everything: Alzheimer, Parkinson's, so on and so forth, diabetes, IBS, cancer, IBD, and many youngsters are interested. You will find that one example, Mark Smith of Open Biome. Then you have Bernard Ole of Vedanta Biosciences. So these are the people who try to make microbiome as a successful business. So it's an emerging sector, lot of opportunities to do microbiome business, and the business is now slowly moving from crude fecal microbiota to specific consortia, to isolates, to specific products. So it's a very, very upcoming business. Coming back to our own country, we have tremendous geographic, dietary, and ethnic diversity, and hence it offers unique opportunity to study microbiome. And we have been studying microbiome in health and disease, obesity, diabetes, celiac disease, inflammatory bowel disease, and mother-infant gut microbiota, age-related differences, and genome sequencing. I'll not bother you with all the details of this study. I'll just show you here that our studies and also the studies done by others indicate that microbes in the gut of our people are different from those in Western world, other Asian countries, and Africa. So these are smaller studies, definitely making a demand for large national level study on microbiome of Indian population living in different biogeographical regions and with different diets. So that's why this Department of Biotechnology has initiated this project. Currently in its shortened version, gut microbiome of individuals from 4 different biogeographic regions belonging to 17 communities is going to be studied along with their all dietary and other information. These are the participating institutes for this study, and with this study one is expected to come up. It is also linked to human genome studies, and eventually one is expected to come up with India specific health solutions using microbiome. This is the final goal of this study. And now finally, for the benefit of local population, I have just summarized what I say in local language. You have to excuse me if I had done any mistakes, because since my representative from Bengal was not in the lab today, he was unwell and he was going to come only in the afternoon, I have used Google Translator to make this. So what it essentially says is why the microbiome is important. It affects our physiology and health, and it gets changed during the disease. And it is important then to maintain a healthy microbiome. And how do we maintain a healthy microbiome? You don't consume any junk food. By junk food, I mean the food rich in high sugar, fat, and low in fiber. Don't consume that food. Say no to aerated drinks also, because they affect your microbiome in a bad way. Have fresh fruits, traditional food, and traditionally fermented food. Lot of such foods you should increase, and that way you'll maintain a healthy microbiome and health. So these are my collaborators. These are my students who have worked on with me on variety of projects. My collaborators from different part of the country with different expertise like diabetes, maternal health, anaerobes, anthropology. And thank you very much for your patience despite all these technical glitches. Thank you very much.
Thank you very much, sir. This was really elaborated, and we have some questions. I can ask you one question from my side. Sir, this may be very naive question for you and Dr. Souvik, but there must have been some studies done where microbiome has been studied for the twins if they have departed in and situated in two different geographical regions. So if that kind of studies have been done to show that the microbiome is the environmental related to the environment rather than to the host genome.
Yeah. So that is a question which is often asked. But in India, I tried to do this study, but I could not do it. And there are no reported studies on twins from India. But from other places, there are studies on twins. But again, the twins grown in two different environments, such studies have not been done. But environmental factors also do play a role in modulating your microbiome. Yes.
Okay. So yeah, this kind of study, if it is done in India, then probably we will be able to link a lot of
study in India in twins living in two different environments. I have twin, but somehow we could not do the study. That one of the twins staying in Pune and the other one staying somewhere else, but we could not complete the study.
Okay. So thank you, sir. And to take this program further, sir, do you have any question or comment?
No, I mean, it's a fantastic talk. And as always, Jogesh always actually, puts things in the perspective. And so is Shovik did a very great job. I'm sure the students and the other people have really enjoyed the talk. Thanks for taking the trouble, Jogeshji. I know you are so busy.
Thank you. No, sir, it's a pleasure.
For the sake of the students, I think we need to actually somehow manage this time and go ahead. Now, I think Sri Prasad wants to mention something. Yeah, Sri Prasad? You can actually, over to Sandeep. Yeah. Thank you all.
Thank you, sir. So, Mr. Sri Prasad will be introducing you, introducing all of us with the Viva program, the IISF association with Viva, and so over to Mr. Sri Prasad.
Thank you, Sandeep ji. So I'm very happy to see this is a very special occasion, and here today we listened very beautiful two lectures. So after these two beautiful lectures, if I am talking something more, it is not good. But आज ये इंडियन नेशनल साइंस फेस्टिवल के बारे में कुछ अब सभी के साथ ऐसे कुछ बोलना ये तो मेरा कर्तव्य है. So that I have to do. तो ये इंडियन नेशनल साइंस फेस्टिवल हम लोग सभी को ऐसे ये फेस्टिवल के बारे में मालूम है. 2015 से हम लोग ये प्रोग्राम शुरू किया. तो what I have to say here of what is the importance of this festival, what is Indian National Science Festival, and what is the philosophy of this festival that I would like to share with all of you. तो ये हमारे देश में ऐसे बहुत सारे प्रोग्राम साइंस के हम बहुत सारे वैज्ञानिक लोग हैं, इवन इफ इट इज ए माइनॉरिटी, फिर भी ये बहुत सारे प्रोग्राम्स इस बड़ा देश में होते रहते हैं. विज्ञान का ऐसे प्रचलन करने के लिए बहुत सारे ऑर्गेनाइजेशन हैं, बहुत सारे प्रोग्राम्स हैं. सो ये सब प्रोग्राम, ये सब इवेंट्स और सेमिनार्स, कॉन्फ्रेंस, ये सब हमारा पूरा समाज के अंदर तक जा रहे हैं क्या? ऐसे एक सोच से हम लोग ये इंडियन नेशनल साइंस फेस्टिवल का कॉन्सेप्ट फॉर्म किया. तो इंडियन नेशनल साइंस फेस्टिवल ने हम लोग ऐसे सोचने के टाइम हमारा मन में ऐसे है, दिस फेस्टिवल शुड रीच द एंटायर कम्युनिटी ऑफ आवर नेशन. ऐसे कोई व्यक्ति ऐसे एक अपर लेयर और वेरी एजुकेटेड और साइंटिस्ट ऐसे एक लेयर में ऐसे इसका लिमिटेशन नहीं होना चाहिए. दिस फेस्टिवल शुड रीच अप टू द एकदम बॉटम लेवल ऑफ द सोसाइटी. कोई स्टूडेंट, कोई फार्मर्स और कोई व्यक्ति, दोस आर नॉट डायरेक्टली कनेक्टेड विद साइंस, बट इवन देन दे हैव टू बी पार्ट ऑफ दिस साइंस. बिकॉज ऐसे हमारा पूरा विश्व में कोई नेशन ऐसे विदाउट साइंस का डेवलपमेंट के बिना ऐसे डेवलपमेंट नहीं हो. वेयर द साइंस इज डेवलप्ड, दैट कंट्री इज ओनली डेवलप्ड. सो दिस साइंस का ये रिजल्ट का और इसका फ्रूट ऑफ दिस रिसर्च दैट शुड रीच टू द एंटायर सोसाइटी. तो ऐसे एक विचार से इंटरनेशनल साइंस फेस्टिवल हम लोग आयोजित किया. तो इसमें हम लोग देखने के समय उसमें एक फेस्टिवल का शब्द भी आता है. What is the connection between India, science and festival? So that is touching the philosophy of this program. It should be a celebrative mode. ऐसे हमारे देश में ऐसे बहुत सारे व्यक्ति और स्टूडेंट्स लाइफ में हम लोग एंटर करने के समय वी आर फीलिंग दैट साइंस इज अ वेरी डिफिकल्ट सब्जेक्ट. तो एक कठिनाई एक भाव है साइंस को, इसको दूरी करना है, उसको एक आनंद का रूप में हम लोग लेकर आना है. When we are celebrating anything, when we are watching that anything with a mode of in the mode of festival or in the feel of आनंद के भाव में हम लोग देखने के टाइम it will touch our heart or we will enter into that. So ये आनंद का भाव और फेस्टिवल का मूड हम लोग ये विज्ञान का क्षेत्र में लेकर आना है. So that is the concept behind this Indian National Science Festival. तो हम लोग ये 2015 में IIT दिल्ली में ये इंडियन नेशनल साइंस फेस्टिवल का फर्स्ट प्रोग्राम हम लोग आयोजन किया. उस समय लगभग 10 to 15 प्रोग्राम्स था. यंग साइंटिस्ट कॉन्फ्रेंस था, एक मेगा साइंस एग्जीबिशन था, ऐसे बहुत कम प्रोग्राम्स था. अराउंड 10,000 के नीचे रजिस्टर्ड डेलीगेट्स था उस प्रोग्राम में. तो उससे 2015 से हम लोग उसके बाद फिर एक बार दिल्ली में आयोजन किया NPL दिल्ली में, और उसके बाद साउथ इंडिया में ये प्रोग्राम आयोजन किया चेन्नई IIT और अन्ना यूनिवर्सिटी के साथ, और उसके बाद नॉर्थ इंडिया में फिर से आया लखनऊ में ये प्रोग्राम किया. और लास्ट ईयर हम सब लोगों को एक सौभाग्य मिला, मेनली बंगाल का साइंटिस्ट और बाकी सब लोगों को एक सौभाग्य मिला हम लोग को यह प्रोग्राम कोलकाता में आयोजन करने के लिए. तो इससे 2015 से 2019 और इस साल 2020 के समय आने के टाइम इस फेस्टिवल इज गोइंग टू मच बिगर दैन when we organized in the first IISF in Delhi IIT. Last year in IISF 2019 in Kolkata, we conducted this program with 28 events. In the 28 events, around 20,000 registered delegates in different programs. And this year, with the direction with our Honorable Minister Harshvardhan ji, in the year of 2020, we are conducting this program with 40 events. So in this 40 events is touching each and every corner of our Bharat, each and every session of our society. ऐसे स्टूडेंट्स के एक 6th स्टैंडर्ड से पढ़ने क्लास में अटेंड करने वाले स्टूडेंट्स से रिसर्च स्कॉलर, यंग साइंटिस्ट कॉन्फ्रेंस में अटेंड करने वाले रिसर्च स्कॉलर तक, और ऐसे हमारा खेती में काम करने वाला फार्मर्स से ऐसे हमारा एग्रीकल्चरल यूनिवर्सिटी और रिसर्च इंस्टीट्यूट में काम करने वाले साइंटिस्ट तक, ऐसे हमारा साइंस के ऊपर लिखने वाले व्यक्ति तक, परफॉर्मिंग आर्ट्स करने वाला आर्टिस्ट तक, ऐसे समाज का विभिन्न भाव से अलग अलग व्यक्तियों के हम लोग एक ही अम्ब्रेला के नीचे में लेकर आ रहे हैं. उसमें हम सब लोग एक साथ आ रहे हैं, सब उसके कनेक्टिंग पॉइंट है विज्ञान. साइंस के बारे में डिस्कस करने के लिए, साइंस के बारे में पॉलिसी मेकिंग वाला व्यक्तियों के साथ हमें लास्ट ईयर हम लोग का ऐसे सौभाग्य मिला. ये विश्व बंगला कन्वेंशन सेंटर में हम सब लोग देखा भारत का सभी स्टेट का साइंस टेक्नोलॉजी मिनिस्ट्री, दोस हु आर द पॉलिसी मेकर्स ऑफ साइंस, ऑल दे केम टुगेदर. एंड उसके साथ साथ भारत का कोने कोने से स्टूडेंट्स, ऐसे हमारे MP लोग ऐसे अडॉप्ट किया आदर्श विलेज, भारत के एकदम बैकवर्ड विलेज से. ये बैकवर्ड विलेज से स्टूडेंट्स कोलकाता में आया, अराउंड 2500 स्टूडेंट्स कोलकाता में आया. ये सब स्टूडेंट एक साथ हमारे साथ था, साइंटिस्ट लोग ऐसे स्टूडेंट्स के साथ इंटरेक्ट किया. सो ऐसे एक बहुत बड़ा ऐसे एक सौभाग्य ये स्टूडेंट्स को मिला. बहुत सारे स्टूडेंट के साथ बात करने के टाइम बहुत सारे स्टूडेंट फर्स्ट टाइम ट्रेन में इसका ट्रैवल करते हैं. ऐसे स्टूडेंट के एक मौका मिल रहा है ऐसे एक प्रोग्राम में आने के लिए, और सभी ये विज्ञान के बारे में इतना एक उसका नॉलेज मिलने के लिए they are igniting with that moment. When one spark is happening over there, that student will be turned to be next Kalam. So this festival is trying to reach out to the village or any student, any class and session of our society. So that is the philosophy of this International Science Festival. We have to unite all our nation to this one subject, that is Vigyan. So that is a लेकर हम लोग इसका आयोजन कर रहे हैं. तो इस साल भी ऐसे इस पैंडेमिक सिचुएशन में भी ऐसे हम लोग इसके नहीं छोड़ सकते, ये इंटरनेशनल साइंस फेस्टिवल आगे लेंगे. So this year we are coming with a new unique platform, that is in a virtual mode we are conducting this program. 40 events with lakhs of participants, those who are watching the program or those who are attending the program. So lakhs of people are going to attend this program and watching this program. And even this Indian National Science Festival itself is a Guinness World going to attend the Guinness World Record in this 22nd to 25th. In this limited days, maximum people are attending a science program. So that is the one Guinness World Record event. And around 25,000 students are at a time they are going to attending an online class. So it is a variety of programs and unique programs we are organizing in this Indian National Science Festival. So I say ये आज का ये कार्यक्रम through this Vigyan Yatra and outreach program, each and every districts of our Bharat or wherever our science related institute or research centers are situated, everywhere we are celebrating. Today morning I have attended the program in Lucknow, afternoon here I am in Kalyani. So through this virtual platform we are going everywhere. So everywhere this message is going. India have a rich, proud heritage in science, and now also we are behind no one. So that message we have to reach everywhere through this program. Is a part of our agenda, and agenda means our motto of this International Science Festival, that is Atmanirbhar Bharat through Vigyan, and that is for global welfare. So that is the main objective of this year, or the theme of this year International Science Festival, Science for Self-Reliant India, and that is for global welfare. That is our theme. So through this science festival or through the development of science, हमारा देश एक self-reliant country होना चाहिए. And that when we are reaching into that goal, our country will be in the position to give a global welfare. So that is our aim. So to reach out to that aim, and we have to organize this type of program to inclusive of everyone in this society in our the nation. So this is a small steps, and we will progress more next year. I think after this pandemic era, we will go to very big program. More than 40 events we will come together in physical mode and we will organize. So this year in this virtual platform, we will all assemble together on 22nd to 25th. So this message we have to reach everyone. This message we will pass to everyone. So ऐसे आज एक प्रोग्राम ऑर्गेनाइज करके ये मैसेज सभी के सामने प्रस्तुत करने के लिए NIBMG ने ऐसे कोशिश किया. तो इसके लिए मैं बहुत आभार हूँ आप सभी वैज्ञानिक लोगों को, और सभी को मेरा आभार यहाँ व्यक्त करके मैं समाप्त करूँ. धन्यवाद.
बहुत धन्यवाद मिस्टर श्री प्रसाद. I request Professor Soumitra Das to add any comment if he wants to have here.
No, actually, I mean, what Sri Prasad ji has pointed out, that we need to really nurture the budding talents. You know, the students are actually the future of our country, and I liked the spirit that we are behind no one. So India has the capability. Indian students have the potential to perform, and you know, the Indians only go and then perform in the foreign countries, and they are actually we are making them proud for those discoveries. I think if we can retain our talent in India, and we can actually attract them and give them infrastructure, facilities, all the avenues that they need, so that actually they can work for the India. And then I think other people will probably attract foreign people to come to India and get this learning. We recently had a Boivov summit where actually NIBMG actually participated in it, and we actually conducted one session, vertical. And there also the motto is to bring in the international scientists to India and then try to see whether we can collectively address something. So all this exposure has to be somehow communicated to the talents, the budding talents, so that they remain attracted to science and technology. It's very, very important. These days, there is no dearth of destruction. So if we somehow prevent them from distracted, remain focused on their career, I'm sure, I think we'll be able to get a lot of good scientists from these budding talents. And I really encourage this kind of things at NIBMG. We all faculty are actually very, very enthusiastic to train young talents, and we try to help them so that they can grow. We are actually ready to handhold people who needs help. Actually, we have a lot of outreach program. Professor Arindam Maitro and Professor Sharmila Sengupta, they have been doing it in the Northeast, so that people from Northeast area, they can come and then work here at NIBMG, get this exposure. So whoever, as Prashadji was mentioning, whoever needs an exposure, we need to spot the talent. Spotting the talent is important. Then once you spot it, give it to us, we'll make sure that these talents are nurtured and actually they can flourish. That is very important. And these programs, these outreach programs and IISF is one such where actually we can spot the talents. You know, I was in Lucknow and then I remember there are 5,000 students, they were isolating DNA in one shot. And, you know, this is essentially the enthusiasm that I have seen there. You know, those are the things that we need to somehow spread it out. And with this spirit, I think I just want to convey the message on behalf of all of our faculty colleagues. Professor Parthapratim Majumdar, with a great vision, started this institute. When he thought about this, the concept of biomedical genomics, that was a completely new concept. And look, after 10 years, Government of India, all over India, now biomedical genomics is actually now the top, because we know that without knowing the genomics, we cannot go for a precision medicine. And that's how, you know, essentially we have to think ahead of time. I'm sure all our faculty colleagues will keep thinking out of the box and try to see something, and then we'll try to fulfill Professor Partha Majumdar's dream. And we try to move ahead and try to see how our young talents can join our bandwagon and actually take it forward. And I think that is very important. And I'm sure, I don't know how many people participated today, but I wish the moment we have vaccines in place and people are free to move, I think we'll be happy to accommodate students in the campus and let them come visit. We have a beautiful museum. Again, Professor Partha Majumdar has made a beautiful museum in our campus. So, you know, that museum is something where we train like how the evolutions and the discoveries of genomics, so they can have a first-hand experience. We'll arrange those things. And I'm sure, and I think Vigyan Bharati should just continue this effort. And this is definitely going to help us to nurture and actually go ahead to get much more and then larger number of scientists in future who will take care of India. We won't really be behind no one. I like that spirit. Thank you. And I think Alpana will give the vote of thanks, right, Sandeep?
Yeah, yes, sir. So thank you, Mr. Sri Prasad and Professor Das for telling us the importance of going out and telling people that what we do and what we should be doing. Our microbiome talk has intrigued young minds, and we are getting question on YouTube. So we do not have a lot of time, but I can summarize one or two questions if Dr. Soche allows, and Dr. Mukherjee can also take the question.
That'd be good.
So yeah, this may be very naive question coming from school students maybe. The question is that if the animal body has microbiome, and is it different from one animal to the other animal, and whether we can take this out, whether we can remove it from the body and study it.
Yes. So can I answer this question?
Yes, sir. Yes, sir.
Yeah. So the answer to that question is yes. Like human beings, even animals have a microbiome which is specific to them. And I gave you some examples as to how in the insect world, nature has ensured passage of microbiome from mother to infant. So yes, I mean, animals also have their own microbiome, and lot of microbiome studies have been done on animals also. It's of course important animals like cattle and some other animal studies are done, but many unique, interesting animals like camel and others also microbiome studies have been done.
Thank you, sir. Probably this person will take this thing further, and maybe he will be someday with us doing microbiome research. So, yes. I will request now Dr. Alpana Dave. She is our academic coordinator at NIBMG to put the vote of thanks for Dr. Alpana.
Thank you, Dr. Sandeep. And this session was really interesting. It was great to hear both the talks. So on behalf of NIBMG, I would like to thank first and foremost IISF for giving us this opportunity to organize this session. And I'd like to thank Dr. Rita Bhattacharya for joining in and Mr. Sriprasad for briefing us the importance of IISF. And I hope that this time the event is going online and that we'll have a lot more participation with students being able to join from all across India and being able to attend whichever session that they'd like to. Sincere thanks to both the speakers, Professor Shauche and Dr. Mukherjee, for simplifying microbiome for us, putting it in perspective, and also sharing some really exciting results from your ongoing research. We do look forward to having some more India-specific microbiome studies and some the importance of our diet on microbiome. It will be really exciting to know all those things. We are grateful to Professor Soumya Das, for the director NIBMG, for his constant support and encouragement. A very big thank you to the organizing team, Dr. Sandeep Singh, Dr. Alindam Maitra, Mr. Vikram Roy and his IT team for their efforts in putting together this event and broadcasting it on YouTube. I also extend my thanks to the NIBMG faculty and staff for their kind cooperation for this event. And last but not the least, all the audience who have managed to join in and have attended this event. I hope that you all have benefited and have gained some new knowledge about microbiome, and you'll be interested to know more in the coming years. And also that you'll be excited to participate in IISF, which is organized from the 22nd to 25th of December. It's an online event this time, and as the video was shown in the beginning of our session, you can always go onto the website to get some more information. Thank you, everyone, and we look forward to the IISF main event now.
Thank you, everyone. So this has brought to the end of this session, event, and we look forward to meet again on 22nd to 25th for main event of IISF. Thank you.
Thank you.